Hello, everybody, and welcome to today's webinar, Primary Container Closures Qualification. I'm Tim Wright, Editor of Contract Pharma, and our webcast today is being sponsored by Nelson Labs. They're a global leader in microbiological and analytical chemistry testing and advisory services for the med device and pharma industries. Nelson Labs supports customers from initial product development and sterilization validation through regulatory approval and ongoing product testing for sterility, safety, and quality assurance. You can visit them online by clicking their logo there at the right-hand side of your page at any time during today's session. Our speakers today are Ario Nicopour and Shiri Hector. Ario is Vice President of Nelson Labs' Global Pharma segment. He's been with Nelson Labs since 2021 and has over 30 years experience in the pharma industry with expertise in analytical research and development, quality control, stability studies, and associated cGMP regulatory requirements. And Shiri is the Senior Manager of Scientific and Technical Services at Nelson Labs. An analytical chemistry testing expert, Shiri currently is part of Nelson's Global Pharma segment team, supporting analytical testing of pharma products at Nelson's new Pharma Center of Excellence in Itasca, Illinois. And during today's session, Ario and Shiri will provide a high-level introduction to primary packaging qualification in the pharma industry. We will be accepting your questions throughout the talk. You can send those in using the box at the bottom of your screen, and questions will be answered after the presentation in a brief Q&A. And to enhance your viewing experience, feel free to customize your window display. You can move the windows around by clicking the top header of each window and dragging them around. And you can also activate the closed caption function by clicking on the CC button in your media player. You can also share your reaction to today's content with the emojis provided, and today's speakers will be able to see your reactions in real time. And a PDF of today's session is not available for download today, but if you are interested in receiving it, we'll give you both Ario and Shiri's contact info towards the end of today's talk, and you can reach out to them directly to get that. And lastly, if you're experiencing any technical difficulties, you can type those into the question box as well, and someone on the back end here will help you out. At this point, I will turn it over to Ario. I will drop off and pop back on at the end for some Q&A. So it's all yours, Ario.
Good day. As Tim mentioned, we're going to discuss the requirement for primary container closure qualification, and the focus is on parenteral and ophthalmic drug delivery system. The outline is we basically going to cover what is container closure system, some definition, regulatory requirements, and suitability of container closure with focus on compatibility studies, which I will cover all those three items. Then my colleague, Shiri Hector, will focus on quality control requirement for primary container closure systems. What is container closure system, or how it's defined? Container closure system consists of really three parts. Number one, material of constructions. You need to know what is material of constructions, and based on that, you need to make sure to do your risk assessment. Then the primary container closure itself, and then the final packaging, which can consist of a secondary container closure and the box and label included. So what is secondary container closure? Is the outer packaging that holds and protects the primary container. It typically includes carton, boxes, or shrink wraps used to group multiple primary packaging together. And in some cases, secondary packaging becomes very important, and especially for the semi-permeable container closures, such as low-density polyethylene for inhalation solution and ophthalmic solutions. Key function of secondary packaging is protection of primary container during the storage and shipping, labeling, tamper evidence, and aggregation for supply chain traceability and distribution. Example would be serialization. As far as regulatory requirement, it is stated in the FDA 21 CFR 211.94. What are the requirements for a primary and secondary container closure, and also there are relevant information in various other guidance or, for instance, technical report from a PDA, technical report 27, and most recently there has been a guidance from FDA which was published in 2024, and that's in regard to changes to a container closure in support of ANDA, NDA, BLA. But our talk is mostly based on the guidance established in 1999 for container closure system by FDA. And so most of the talk is, as far as the requirement, is based on that guidance, which is still relevant today. Composition of container closure system. The composition of container closure system it can vary, because there are sometimes you have a plastic container closure, as I mentioned, like a low density polyethylene, but most of the time glass, and then you have stopper system, or delivery system, which can consist of multiple components, such as elastomeric components, plastic, and even metals. Then also the secondary packaging material needs to be well understood in the final shipment box. Selection of container closure system, you need to take following into consideration. First of all, must protect the formulation from environmental conditions, must not be reactive with the formulation, and must not alter the taste, odor, appearance, and color of the formulation, must be safe. In addition to that, it needs to ensure tight seal that prevents microbial ingress, gas exchange, loss of contaminant, loss of content. Suitability of primary container closure. At minimum, 3 things needs to be taken into consideration: safety of container closure, protection, and compatibility of container closure. But in case of delivery systems, such as pre-filled syringe, auto-injector, cartridges, or meter dose, for example, meter dose systems, you need to also look at the performance of the primary container closure. Then, in addition to that, you need to also establish the quality control test requirements for the container closure. Primary container closure system for a parenteral and Ophthalmics, minimum, they need to definitely ensure the stability of the product or protection, safety, compatibility, and performance. Key requirements and aspects of qualification. What do you need to do for each one? For protection, for instance, you need to make sure it's protected from environmental moisture, atmospheric gases, so you prevent, for instance, auto oxidation of the drug. Lights, temperature fluctuation, how it impacts the drug product through shelf life. Particles, foreign particle matters, and microorganisms. Safety, really, you need to understand what is your container closure system made of. And based on that, you can do your safety assessment early on by, for example, looking at the open DMF for your container closure and for each of the component, that can give you some level of confidence. And then also you need to do studies such as extractable leachables to determine if anything can leach into your product. Compatibility. The container closure must be chemically and physically compatible with drug product, preventing any unwanted reaction or interaction that could compromise drug quality and safety. And I will talk about compatibility in more detail later. And the performance of the container closure, of course, the CCI or container closure integrity is very important to throughout the life cycle of drug, but there are other aspects of the performance you need to look into, which we'll discuss later. Container closure integrity test. This is a test which is done by various techniques, deterministic method and probabilistic methods. And deterministic methods are favored, such as mass extraction, for instance. That's something we use at our laboratory. Vacuum decay, laser-based methods. Those are some of the methods. And then stability testing ensures that the product, it is stable and not reacting to a container closure throughout the shelf life. The change management. Any changes to the container closure system, including material design or manufacturing process, should be thoroughly evaluated and reported in accordance with the regulatory guidance. In the beginning, I mentioned that there is a new guidance established by FDA on 2024 about those particular changes and how you need to address it. In essence, container closure qualification is a multifaceted process that ensures packaging system is suitable for intended drug product, thereby protecting quality, safety, and efficacy throughout the shelf life or life cycle of the product. This table is actually from the FDA packaging guidance established in 1999 and today still relevant. It talks about degree of concern associated with route of administration. For instance, inhalation, parenteral products, these are really highest degree of concern. And the likelihood of packaging component interacting with your formulation, specifically primary container closure. So as you can see, I've circled both injectable product in form of solution or suspension, and also dry powder inhaler. So those two formulation, there is you need to do a high level of scrutiny to determine if there is any interaction between your formulation and your primary container closure, or even in some cases secondary container closure. However, even though in this table says certain things such as oral tablets or oral capsules are immune from any interaction or leachables, that's really not true. In past decade, we have found out that some material can leach into the even oral solid dose. For example, in case of blister packs, there has been report of leachable from foil, overwrap foil or foil laminate on the PVC casing, which certain chemicals have migrated into the product. This table is actually a summary table from Annex 1. And Annex 1 really touched bases on several compatibility studies: chemical compatibility, physical compatibility, microbial compatibility, and functional compatibility. Then also addresses the labeling and storage conditions. For chemical compatibility, there are leachable studies, which is very important. And fortunately, we have relevant guidelines today in both ICH and also USP 1663, which is for extractable, and 1664 for leachable studies. You need to evaluate those. Then also you need to have other methodologies in place. For small molecule assay and related method, stability indicating method can be a very important factor. However, when it gets to biologics, you need to have a lot more methodologies in place to look at different attributes. For instance, aggregation of the protein, denaturation, surface absorption, and so on. So you need multiple methodologies for biotechnology-derived product to address the chemical compatibility, whereas in small molecule methodologies such as pH, osmolality, assay-related can really reveal a lot of information for you. Then physical compatibility will really determine the things such as closure torque, container deformation, absorption or precipitation of the product. And this can definitely be monitored during course of the stability, either R&D stability studies or stability studies in support of shelf life. And microbial compatibility verifies still barrier over shelf life. This is addressed in a USP guidance 1207. And functional compatibility, if you have a delivery system, you need to look into dose delivery, sterility, needle safety, product identification, and user safety. Then also, as I mentioned, you need to look at the labeling and storage condition, monitor chemical and physical changes for those. Step for qualification of a container closure system. Component characterization. This is basically USP general chapters, which my colleague Sherry is going to talk about all of those specifically and each requirement. Then performance. Safety, as I mentioned, extractable, leachable is one of those important methods you need to look into or process. Then indirect food additives in reference, biocompatibility, particles, absence of adverse reaction trend to components. Then for compatibility itself, permeation. This is very important when you're dealing with a semi-permeable container closure. Leaching again, sorption, chemical reactivity, modification, particle formulation, drug product precipitation, confirm the functionality of delivery. And for protection, you need to do studies such as light study. You need to look at the CCI for microbial and permeation. For compatibility study, again, you need to really define your container type. What is it? Is it just violent stopper, or it's a pre-filled syringe, auto injector, ampule? And based on that, you need to design your studies. Then the material of construction is also very important. So you have to have a good knowledge of your material of construction. And if possible, you need to ask for open DMF from your supplier. Characteristic of formulation. What type of formulation do you have? Is it suspension, liquid? Is it lyophilized? And so on. And based on that, you need to look at the importance attributes such as osmolality, viscosity, ionic strength, and pH. Then stability profile and route of administration. It also comes, it becomes very important. Is it IV, IM, or subcutaneous? Determine the study condition. What do you want to really evaluate under realistic and stress condition? Temperature, moisture, light, and the duration. Define your methodologies. As I mentioned, for small molecule is quite simple, simpler than biologics. Biologic, you need to really identify additional methodologies to look at different attributes. But for small molecule assay and purity method, it really is very vital. pH, osmolality, viscosity, particulate matter, and moisture, if it's lyophilized product, those are important methodologies to establish in advance. And control strategy, you need to really have an incoming container closure system material specification, integrity testing, visual inspection, and functionality testing. And of course, evaluate risk-based design. For the platform type of formulation or platform delivery system, leverage the existing platform data or supplier DMF, but always verify compatibility with your specific formulation. The compatibility with formulation, you really need to look at three factors: a chemical compatibility, functional compatibility, and physical compatibility. For the physical compatibility, one thing which becomes very important, it's really absorption study and adsorption study. Adsorption study is binding to the surface. So in advance, you need to understand if your drug of choice or the components, they have any affinity for those surfaces. It can be elastomeric, your glass, or plastics. And absorption is penetrating the bulk material to bulk material. You need to design those studies and also have prior knowledge of your components and also the formulation itself. You need to also look at the appearance. Color change and clarity gives you a lot of good indication. And glass delamination, that also should be a subject to study. Discoloration, surface residues, these are all very important information to gather and also studies to do. For chemical, as I have mentioned, you need to definitely do assay related and also extractable, leachable. And for functional, there are a series of tests you need to look at, and that's based on your primary container closure. Is it a delivery system or just a container closure? And based on that, you design the studies you need to do. So in summary, you need to do really following. Component characterization is the material. You need to understand the material composition, physical and chemical properties, chemical reactivity and leachability, microbiological and particulate cleanness, and also sterilization compatibility. That is very important. And sometimes that's ignored because people don't have the knowledge if this material has been sterilized or not. And if it's sterilized, in certain cases, you need to look at the residues. For example, if noxalizer is used, you need to look for a residual nitrate and nitrite. If it's EO, ethylene oxide, which really that's not used commonly in pharmaceutical, but you need to look at residues for those. But something like gamma, of course, there is no residues. However, sterilization can cause other serious issues and can change the components, or in case of if product is sterilized, it can cause degradation of the product. Then for functional testing, you need to look at the compression force penetrability, closeness of fit, seal integrity. As I mentioned, you look at those with a deterministic or probabilistic method, such as helium leak, dye ingress, mass extraction, or other techniques. Resealability. This is basically stopper puncture testing. Break loose and extrusion force for pre-filled syringes. Delivery force, gliding force, break loose force. Then extractable, leachable, we touch base on that extensively, so there is no need to talk about it again. And compatibility itself, evaluate chemical and physical interaction between formulation and container closure system. Ensure no leachable, extractables, or adsorption affects the drug product or absorption. Confirm closure integrity and protection throughout shelf life. Demonstrate that container closure does not affect potency, purity, pH, osmolality. And then product contact surface evaluation. Evaluating product contact surface of container closure is a critical part of drug product development, especially for parenteral and biologic parenteral. This ensures that the material in direct contact with the drug do not adversely affect safety, identity, strength, quality, and purity over the product shelf life. Then you need to also look at the surface interaction and adsorption evaluation. Microbial barrier evaluation is basically CCIT supports that. And as I mentioned, there are various techniques to follow. And stability of drug product throughout shelf life, ICHQ1, you can follow that and also determine what zone you need to do the study for. And based on those, you determine your conditions and duration of the study. And qualification of manufacturing process, this is the exercise you need to do with your manufacturing people in advance when you are selecting your container closure to make sure your container closure is compatible with the manufacturing processes. Now I'm going to hand it to my colleague, Sherry. Thank you so much for your attention.
Thank you, Aryo. I will talk about the container closure testing requirements from the compendial testing. And really understanding this requirement is crucial for compliance and also to ensure the quality of the packaging system. USP has various container closure chapters. You can see here, they have chapters for elastomeric, glass, and packaging. And these are the EP chapters relative to container closures. As you can see in the slide, the EP chapters are divided a little differently than the USP. They're divided by material and not by container type like the USP. And if you want to do a quick comparison between the EP chapters and the USP chapters, you can see here the EP chapters, the material and the USP equivalent chapters. For example, if we look at EP 3.2.9, the rubber closure, the USP equivalent is USP 381, which is the elastomeric closure. We can also see that the key testing requirements are similar between the USP and the EP. However, they're not fully harmonized. So then when selecting the test requirement, we have to review both compendia carefully to decide what test we need to perform. USP 660 glass containers talk about 3 types of glass that are most commonly used. Type 1, which is borosilicate glass. It has very high hydrolytic resistance, and that is why it's used for biologic, injectables, and sensitive drug to impurities. And then type 3 would be, for instance, the one that we can use for more stable molecule, oral, and liquids. And then this is a table that's taken from USP 660. And it has the performance types of glass and the test required for each glass. And just as a side note, the EP equivalent chapter for the glass container is 3.2.1. And traditionally, the EP and the USP had similar aspects of the testing, but USP revised their chapter in 2023. And they moved from composition-based classification of glass type to performance-based approach. This change was driven by emerge of new glass compositions like aluminum silicate glass, and some recommendation to allow flexibility and innovation in glass packaging. So now USP defines type 1, 2, and 3 by performance, as you can see in the table here, not strictly by composition. Moving to the plastic chapter, the original chapter of plastic and material for construction is USP 661. And there are new chapters, 661.1 and 661.2, which become official in December 1st, 2025. USP 661.1 deals with the plastic material for construction, and USP 661.2 focuses on the suitability, safety of the complete packaging system. As you can see, there's more enhanced quality and enhanced regulation, because now the chapter addresses the material of construction and then the full package system. The qualification is based on 3-stage approach. The first stage is material assessment, characterization, screening, and selection per USP 661.1. Second stage would be package system assessment and qualification, USP 661.2, and 1663, which is the extractable chapter. And the last stage would be product assessment and qualification, which is USP 661.2, and the leachable chapter. If we want to compare between USP 661 to the new chapter, we immediately see that the new chapters include many more plastics that were originally included in USP 661.1. A lot of them are relating to parenteral products like HDPE, high polyethylene, high density polyethylene, low density polyethylene, polypropylene, PVC, etc. There's also unaddressed materials that are detailed in the USP. And those are plastic packaging that are not detailed in the chapter, but they are, the USP calls them unaddressed materials. And for these specific materials that are not included in the USP, but used in the plastic packaging system, the user will have to develop methods similar to 661.1 and also develop acceptance criteria. The plastic additive test, it's a new test that is added to USP 661 and is to test plastic additives which are antioxidant. They can be phenolic or non-phenolic, and those are additives that are added during the manufacturing process to the plastic to optimize its performance. And the testing for plastic additives is really meant to is designed to control the amount of plastic additive added to to the plastic or material for construction. And it's designed to detect and quantify the antioxidant that can leach from the plastic packaging to the pharmaceutical product. This enables us to ensure that the materials used are safe, compatible, and functional. The testing for plastic additives is not required for oral and topical dosage forms. The technology we use is primarily HPLC for phenolic antioxidant and TLC for non-phenolic antioxidant. When we want to choose the plastic additives that we want to test, we will choose the ones that are detailed in the composition of the plastic. So we need this information in advance from the manufacturers. For the HPLC quantitative test, method feasibility and verification is required. It is a limit test, so normal characteristics would be system suit, specificity, limit of detection, and stability of solutions. And obviously, when introducing new tests, there's challenges. So the challenges that we observed when we were testing for plastic additives by HPLC were mainly unknown impurities and co-eluting peaks. The USP states that only the peaks that caused by the antioxidant should be observable in the sample chromatogram, or maybe the peaks from the blank. We have seen some cases when there is additional peak, unknown peaks that are not related to the plastic additives that are detailed in the composition of the plastic. And those peaks needs to be identified. There is also cases when those peaks co-elute with the reference standards of the plastic additive, which made it hard to integrate and quantify the plastic additives in the sample solutions. And this is why method feasibility and verification is critical to perform before we actually perform the routine test. That's because during method feasibility, we can see the chromatogram of the sample. We can optimize the HPLC parameters to separate peaks. We can see how many unknown impurities are, what the level to determine if the plastic is going to pass and going to conform with the USP requirement for plastic additives. Additional tests that are detailed in USP 661 and are not in the original chapter are related substances tests, and those are specific to the plastic or material for construction. The technology that is commonly used is titration or gas chromatography. And for this also, method feasibility and verification is required from the same reason we detailed before. The benefits of the new chapters, they provide enhanced safety and quality for the packaging materials because we address the material for construction and then the full package system. They provide comprehensive testing for extractable and leachable. They provide implementation of risk-based assessment approach, which can save time and money in testing. And they provide alignment with global regulatory standards. The challenges are, and obviously when we implement in new testing chapters, there's always challenges. The first one is the complexity and scope of testing. As we said, we're introducing new technologies, chromatography that those tests are more complex and they can cause challenges in testing. The cost and resource burden, that's another challenge that labs needs to address or manufacturers needs to address because we're introducing new technologies, we need new SMEs, training, equipment. Interpretation and flexibility of the results. If before we kind of said that the fact that its risk-based approach is actually a benefit, it can also pose a challenge because the interpretation can be flexible and maybe different between manufacturers, and that could be a challenge. Manufacturing and material variabilities, any changes to the resin manufacturing, processing, formulation can cause challenges with the testing or inconsistent results, specifically with tests like TOC, total organic carbon, or UV absorbance. And we have seen some cases where the resin passes USP 661.1, but when it placed in the final packaging, it fails USP 661.2, and those instances needs to be investigated. And the last challenge is challenges with multi-layer materials and unique design. Multi-layer materials can cause challenges with testing per USP 661.1, which is designed mainly for a single polymer. And some of the multi-layer materials, they cannot be separated and tested separately, so that can cause complication in testing. Design like blister packs, which has very, very small cavities, can cause issues because we need to collect a certain amount of volume of extraction to test per USP 661.2. And with blister pack, we would need a lot of blister packs to fill in order to collect the volume that we need. And then the filling is a manual process, and the sealing would be a manual process, and that can also pose risk for contamination. Moving to the elastomeric component in injectable pharmaceutical product, the two main chapters that talks about it are USP 381, which currently has the physical chemical test, some biological reactivity test, and functional test. And USP 382, that's the new USP edition. It will become effective in December 1st of 2025. And that will include many more functional tests and also reference to USP 1207, which is the container closure integrity testing. So if we want to see what USP 382 adds, we can see in this table that it's adding a lot of tests in regards to the functional suitability or functional tests. And we can see that now, if before the testing was only focused on needle and spike access, now we're also required to test the plunger, the tip cap and needle shield, and also perform integrity tests for the closure system. So if I want to summarize the elastomeric component in injectable pharmaceutical products, the USP really had a significant change in 2020, in December 2020, for USP 381. One of the changes is that the heavy metal test that was originally there, which was a qualitative test, was removed. And now they changed the recommendation to have control extraction study, similar to USP 1663, which is the extractable chapter. Another change that was significant was that now the product developers will be responsible for evaluating extractables testing needs and also provide the justification why. And in December 1st of 2025, there was going to be a second change to USP 381. In this change, the functionality tests are going to be removed. Besides the fragmentation test, that's still going to remain in USP 381. And then that's allow implementation of the new chapter 382, which will be the chapter for functional suitability in parenteral products. So as we said, the USP 382 will cover different types of system that's used in injectable pharmaceutical product. And there's another chapter 1382, which is the assessment of elastomeric closure functionality in injectable pharmaceutical packaging. And that will provide guidance on test sample preparation, test population, procedures, ISO standards, data interpretation, and reporting. Some of the challenges with USP 381 and 382 Similar to what we discussed with the new 661 chapters, we're introducing new tests, new more complex tests, and that's always can cause challenge because we need training, resources, and subject matter experts. There is a dual responsibility between the supplier and the end user. So the supplier, the manufacturing of the closure, would need to test it upon manufacturing to ensure compliance of USP 381 and 381, and then the end user will have to test it again after processing or after sterilization that can alter the properties of the closure. And sometimes this dual responsibility can cause gaps in accountability or even redundant testing. Material variability and formulation changes. As we discussed before, any changes to the materials, the design, formulation can cause complexity and changes in the testing and even failure to comply with the chapters. And equipment methodology and changes. We are introducing new equipment and new method that can cause challenges. We have to really carefully choose the equipment that we want to use for the testing to avoid contamination for specific tests, to provide accurate measurements of the testing. So that can be a challenge. So in summary, if we want to summarize the webinar and the key takeaways from container closure qualification, it is important understanding the container closure system, regulatory requirements, ensure safety and efficacy. Material selection is crucial for protecting formulation. Qualification process involve multiple assessment aspects. Compatibility studies, as we've seen, Arya talked about it in detail, are vital for the drug packaging. And the new USP chapter enhance the safety and testing standards. And I will take it back to Tim. Thank you.
All right, great. Thank you so much, Sherry. And thank you, Ario, as well for today's presentation and for sharing your insights with us today. We do have time for some Q&A. So if you do have a question for Sherry or Ario and weren't able to get it in during the talk, now's the time you want to go ahead and type it into the question box at the bottom of your screen there. Are you on camera, Ario, Sherry? Oh, there you guys are. Okay, perfect. All right, let's get into this Q&A here. This attendee would like to know, ask if it's required to evaluate 4 elements detailed in the suitability of primary container closure. I don't know which one of you wants to take that.
I can take that question. Yeah, you definitely need to qualify your container closure system. And you can rely on a platform data if you have previous information, but at minimum, you need to do compatibility because each formulation is different. And also, you need to do some of the physical chemical testing in support of that. So you need to ensure that it is suitable for your specific drug product. And this qualification is a regulatory requirement to ensure product quality, safety, again, and efficacy throughout shelf life.
Okay. Did you want to add anything, Sherry, or are we good with that?
No, I'm good.
All right, let's move along here. And maybe, Sherry, you want to answer this one. What are possible solutions for testing blister packaging per USP 661.2?
So there could be possible approaches to test the blister. One can be manufacture a larger blister like product that can hold more volumes, and that could solve the small volume issue. Another one can submerge the blister pack in the extraction and use it as the worst case scenario. So those are a few that we can think about.
Okay, great. Did you want to add anything, Ario? Or is that she covered
No.
pretty? No? Okay.
Yes, yeah.
All right, we're going to take one more question here before we wrap things up today. And this attendee asks, do we have to test every lot for container closure?
We talked about the testing done by risk-based approach. So really there should be some risk-based approach and justification to how many lots we're testing and why we're testing a specific number of lots. And it also can determine about the qualification that was performed initially. And we can use this as part of justification. So it really varies between the needs.
Okay. Terrific. All right, thank you. All right, everyone, that's going to bring us to the conclusion of today's session. If you did have a question and we weren't able to get to it, Ario and Shiri both have your contact details and they will follow up with you directly shortly after today's broadcast. So thank you again, Ario and Shiri, for a great talk today, for sharing your insights with us. Thank you again to the sponsor of today's session, Nelson Labs. Again, be sure to visit them by clicking their logo at the right-hand side of your screen there before logging off today. And thank you, everyone out there in the audience, for your attendance and participation today. And if you are interested in a PDF of today's talk, again, you can reach out to Shiri and Ario directly, and they will get back to you. And if you aren't able to get to them, feel free to reach out to me as well, and I'll put you in touch. Lastly, this session will be archived for 1 year on our website, contractpharma.com. And you'll also be receiving an email from us shortly when the session's available for on-demand viewing. So thanks very much for joining us, everybody, and have a great rest of your day. Take care.